right. Yeah, rookie time now, so I think we'll get started. Thank you everyone for coming. Today we have a talk from Tianyu, who's a PhD candidate in Possu Huang's group at Stanford, working on generative models for protein structures. Previously, he worked at ProteinQure and studied with Philip Kim and Alan Moses at the University of Toronto, working on DNA mimic proteins and inferring the structure of gene regulatory networks. And today he's got a talk prepared on shapes, which is a framework for evaluating these generative protein design models that addresses some of the key limitations with existing methods. So without further ado, let's give it up for Tianyu.
All right, thank you very much for your interest. Thanks for the introduction and the invitation. So SHAPES stands for Structural and Hierarchical Assessment of Proteins by Embedding Similarity. And this is joint work with an undergrad in our lab, Melissa. So some motivation for this work. And we have a preprint out in the bottom if you want to check it out. The field of protein design has right now revolutionized by using generative models to efficiently sample from this complex data distribution of protein structures. And it turns something that's very difficult to sample from, traditionally requires expensive Monte Carlo sampling, into something that's easy to sample from by fitting these deep generative models. But anecdotally, we know that these models are biased. So for example, from personal experience and also experience from other groups, we know that RF diffusion, for one example, tends to oversample alpha helical structures. The goal of this study is kind of a benchmark of several state-of-the-art generative models for protein structures and understand what kind of distributions they're actually modeling by comparing its sample distribution to a so-called ground truth distribution here, whether it's the CATH database or the protein data bank. So several questions that this study tries to address is what regions of protein structure space are not being covered by generative models. And implicitly, many of these generative models, when you're benchmarking them, there is this key metric which they tend to optimize for, which is designability. And I'll go into a little bit more detail on how designability is defined. How can we quantify and interpret the distributional coverage of protein structure space? Where in this space is there undersampling and where is there oversampling? What limitations does this bias sampling behavior actually place when you give these models to the hands of a protein designer? Ultimately, the goal of design is the ability to impart bespoke functional elements into your structure. So what implications does it have for functional protein design? Ultimately, the scope of the study kind of boils down to the single figure, where you have the purple background representing all of protein structure space. You have the observed subset of that large space, which is the protein data bank. However, the model distribution actually, as it turns out, covers a slightly biased distribution than the observed subset. We've noticed from projections of structure embeddings, which I'll go into detail later, that there are these de novo regions where you do see extrapolation behavior from these generative models. But there's also large regions of the observed space that is missing in coverage. And our claim throughout this is that one of the reasons for this behavior is this optimization for designability causing this shift and this bias. And also, by the way, feel free to ask any questions throughout as well. So sort of to set the stage of what we're considering in this study, generative models are evaluated by designability. So under the traditional generative setting, what we have is a complex data distribution, such as the PDB. You fit a generative model such that it can transform a distribution that's easy to sample from, like a Gaussian, into a distribution that is modeled that approximates your data. Then you try to optimize your loss. And ideally, now when you draw samples from your model distribution, it would be as if you drew samples from the real distribution. However, in generative models for protein structures, there is this designability filter that is imposed on not just the outputs of your models, but also throughout the model development process. You're picking your parameters and your hyperparameters based on what settings give you good designability. So designability and the self-consistency metric, as it can be also called, can be precisely cast into these following steps. Step one, you draw a sample from distribution of structures. Given that sample, you then design sequences. So I'm going to cast this into probabilistic language. In reality, it's not strictly probability distributions, but this is just a nice concrete way of thinking about the problem. So you draw a sample from P2, which is a model of your sequence conditioned on structures. Finally, then you invert this. You take your sequence and then you refold it into this X refold term. So in practice, P1 could be RF diffusion, P2 could be protein MPNN, and P3 could be ESM fold or alpha fold. Then you determine a global optimal alignment of your refolded structure with your initial structure X. That rotation and translation as globally optimal is then applied to one of your structures, and you're comparing the distance between your refolded structure and your original structure, and you're seeing if that distance is under 2 angstroms. Usually that's the default cutoff. In addition, there could be other criteria like the confidence, PLDDT, or PAE. So in some works, you would consider a threshold of 80 or 70. These are essentially high confidence predictions through AlphaFold or ESM fold, or a predicted align error of less than 5. So there are some issues with this setup. In particular, we're unclear what the trust regions of steps 2 and 3 are. We don't know what regions of sequence space or structure space as input to these design models and structure prediction models are reliable. And we don't know what kinds of sequences may be adversarial. There's also a choice of models to use for P2 and P3. The conventional setting is usually protein MPNN for P2, AlphaFold or ESM fold for P3. But there are many, many models that exist for sequence design that model this conditional, and also many models for structure prediction. Steps 4, 5, and 6 implicitly actually penalizes for structural flexibility. So in particular, step 4 determines a singular globally optimal alignment between predicted structure and your original structure. This is in contrast to the fate loss in AlphaFold, where you're actually doing locally optimal alignments per residue. And this can unfairly penalize regions of your structure that are more flexible and more hinge-like in nature. And so achieving this fairly strict threshold of being less than 2 angstroms really can bias you towards more rigid samples and disfavor more flexible samples. So in practice, just an English sample structure, design sequences given your backbone, predict structure of given design sequences. You do a globally optimal alignment. And if your structure is similar up to 0.2 nanometers and that your predictions are highly confident, then that in a way says that your design structure is self-consistent and that is so-called designable. However, a statistic that we found and also confirmed by many other works in the field is that if you pass in through this pipeline native sequences from the PDB, which we know exists in nature, it turns out 40% to 60% of these native structures are not designable. So they fail this pipeline, but the definition of designability is that there exists a sequence for which it folds into that target structure. And we know that these proteins exist. We've observed them with crystallography and other experimental methods. So there's almost this mismatch between this computational proxy and the experimental reality. So the conclusion of this is that generative models of protein structures are biased towards modeling the designable subset of structures and not all native structures. This might be a good thing if the goal is actually to design this hyperstable distribution of proteins for which things are very stable, thermostable, they have high expression, as we've seen with many of the de novo designs with protein MPNN. But the implication of not covering all native structures is that there may be functionalities inherently present in these flexible regions that are not being modeled by the generated by the model distribution. In particular, we quantified the secondary structure distributions, and we noticed that indeed it agrees with anecdotes, where we see the purple background is the cath distribution. So these are structure domains of real proteins that are experimentally observed. And in each of these plots, we're plotting about 21,000 structures sampled from each of these 5 models. So cath is the exact same data set that was used to train Ingram et al.'s paper, which is the original precursor to protein MPNN. And we see that in all of these models we've benchmarked, there is this right upward shift in secondary structures of the sample distribution. So intuitively, that just means you have more beta sheets, more alpha helices, and less loops in the sample structures. You can see that some of these distributions are quite extreme. And in fact, the RF diffusion distribution is all the way down here with the majority of its sequences, of its structures, having mostly alpha content. So analyzing this a little bit further, we're trying to see what exactly does RF diffusion do to an input structure. And this is just one perspective of looking at this. What we did in this experiment is and actually this is kind of already confirmed by Sergey Ovchinnikov in several of his tweets, where you have a base distribution that then you apply partial diffusion using RF diffusion, and you get the resulting distribution from that. So what we did was for every structure that's generated by Propardelli, which is a generative model developed by Possu Huang's group internally, we apply 20 steps of RF diffusion, partial diffusion. So we noise the structure a little bit, and then we denoise it back to data. And we notice this shift in secondary structures elements. So in this middle plot here, you see these streams going from the bottom left corner right upwards in a loss of loop structures, and it's being annealed into both more helical content and more beta sheets. Up here, you see the arrows going downwards like this, where actually the long-range beta contacts are being morphed and slowly becoming more alpha-like. And the result of this is actually a dramatic increase in designability. So this curve here in light blue is Propardelli, where it didn't have the highest designability, but just after applying RF diffusion's partial diffusion, we end up with highly designable structures. And indeed, this could be attributed to the gain in secondary structure, but there are many other factors as well, like it could be actually fixing some clashes in the original structures. But the secondary structure part is definitely quite notable here. To dive into this a bit deeper, we take inspiration from one of the papers from Kevin Yang's group, EvoDiff, where they used protein language model embeddings to look at the sequence distribution between generated sequences and real sequences. We cast that into the protein structure case. Yeah.
Can I have a question about the previous slide?
Yes.
So when you do this analysis, did you check if RF diffusion favors any functional domains when changing the structure to more like alpha fractions?
You mean so it's hard to say because these are all sample structures, so we don't know exactly their functions. And so we could predict those, and there's a slide coming later on, but yeah. Yes.
By any chance, did you try doing partial diffusion on those 40 to 60% of real proteins that are, yeah.
Yeah. No, I haven't done that. I guess, yeah, it doesn't really matter to start from proper deli. Essentially, the goal is to start from some initial distribution that is not perfectly designable. So yeah, I would suspect you would see the same trend. Yes.
Since we're talking about the 40 to 60% of native proteins that aren't designable, I'm wondering if that's obvious computationally, like once you get to the alpha step, or is that something that doesn't become obvious until experimental validation?
Yeah, so the designability part is purely computational. So none of this is experimental at this point. And so by these computational metrics, 40 to 60% do not pass these thresholds. Oh yeah, yeah. So like basically it's like counting the frequency of how many samples have RMSD less than 2 and how many have confidence, the predicted local distance test of above 80. Yeah. Okay. So to go a little deeper into this, we're looking at similarity of representations of protein structures instead of protein sequences, which they did in EvoDiff. For protein structures, there's not a single model that captures an embedding or a representation of your structure. There are various hierarchies of structure. So there are these like local features which are captured by these this fold-seq model, where they only take nearest neighbor amino acid features and they look at their relative distances and angles between one amino acid I and this nearest neighbor J. And that's all it's being featurized. But there are these very global features like these protein domain segmenter features, which are pooled over the entire protein structure. And the output is to predict what cath architecture your input structure belongs to. Then there's something in between where you're capturing sort of local amino acid environment features. And this can be done with message passing or graph neural networks where you're having a node and its nearest neighbors. The information from its nearest neighbors are being aggregated in that form. So one way is protein MPNN, where they take the nearest 48 neighbors. And for ESM3, that encoder inside ESM3, it takes its nearest 16 neighbors. So there are various hierarchies which we consider. And what we're going to do is we're going to featureize a protein structure through each of these embedding types and obtain a per residue embedding for each of the protein structures we have in our sample distribution and in our ground truth distribution. So we get these continuous features, just like protein language models, we mean pool them lengthwise. The protein MPN encoder has 3 encoder layers, so we take features from all 3 of them. Since the first layer looks at its nearest 48 neighbors, after one message passing step, there's 1 extra hop, and so you get a larger field of view as you go down the encoder layers. The ESM3 embeddings are interesting because they use a vector quantized VAE. So what we do is that we take the continuous codebook vectors in the layer immediately prior to the discretization step. So there's a finite number of codebook vectors that you can use for every residue that represents its strict local environment. So we can project these mean pooled vectors into the first 2 principal components, and we can compare the designable subset and the non-designable subset. And it's not entirely obvious from here, from these plots, that there's anything different. And in particular, even if you just look at the secondary structure distributions, the non-designable part is quite similar to the designable part. And so indeed, you could probably design sequences or structures that are quite loopy. And perhaps that the bias towards more secondary structure is an artifact of some of the models. Yes.
Line about embedding types.
Mhm.
So the embedding types actually defines that the radius around neighboring the radius. How do you define how many? Do they have different?
Yes. It really depends on the model. So we don't train new models for this. We take off-the-shelf models, and each of these models have their own way of tuning how many neighbors are optimal for their task. So for protein-MPNN, they did a sweep over how many neighbors they take in, and they found that when they take 48 neighbors, their performance, which is sequence recovery, sort of saturates. So that's sort of their design choice through choosing. Yes.
Or is it variable per line?
These are fixed.
Fixed.
Yes. Okay. So while that these distributions between designable and not designable are not appreciable, we do see through this lens of structure embeddings this pretty striking pattern. So there's several things going on in this plot. One is that so here we're looking at the VQVAE encoder embeddings of ground truth structures on the left and of sample structures on the right. The sample structures indeed have this very notable band, which is so I have in the next slide, I'll show you exactly what these structures look like. But these dots do not show up in the reference distribution. Whereas there's also this region here where it doesn't show up at all in the sample distribution. So there's kind of this undersampling over here, and there's oversampling or generalization over here. And what's quite interesting is that the latent space of this VQVAE is sorted by helicity. Here's just a coloring of the helix content of each of the structures. One subtle but very important point here is that for the reference distribution, we're very careful in choosing what counts as a reference. In the sample structures, we always have continuous chains with no breaks in between. So to prevent any artifacts of unresolved residues going into the representations of our model, we remove all of the experimental structures which have gaps. So any regions that are unresolved and those that are considered poor quality with these cutoffs so we drop anything with resolution worse than 3 angstroms and with a free R factor over 0.25. R free of greater than 0.25 could be considered quite strict for large proteins, but this is quite a reliable metric for overfitting your atomic models to crystallographic data. Okay, so now this is the same plot, but rasterized. And in this plot, you can actually scan the QR code. I think it works on the top right. If you want to look at a collection of sample structures stratified by designable and non-designable, and also ground truth structures. So there's many, many plots that we made. Did it work? Oh, okay. I see. Let me just change that real quick. Okay, I think I updated the permissions. So, okay, great. So this is just two of the slides of the many slides in that raster plot. And I think it's potentially useful to browse through all the distributions. But what you end up seeing is if you look at sort of these dots of what is undersampled and what is generalized, what you end up seeing is that the parts that are being undersampled are these very complex structures. So I can sort of zoom in to this region where these folds are quite complex, involving a lot of loops. Whereas the region that is on the bottom right, strikingly, they're essentially helical bundles. And the generalization happens in this space. What you also end up seeing is what's interesting is that if you just take a random sample of every voxelized region, you see that querying the IDs in the RCSB, all these cyan or bluish background grids are structures that are enzymes or domains of enzymes. So there is a heavy undersampling and underrepresentation of enzymatic structures in the sample distribution. So here's just another perspective. So previously those were the two principal components, and here we're looking at the UMAP projection. The organization is also quite striking. So you see highly idealized beta sheets in one region, highly idealized alpha helices in another region. And the undersampled regions, particularly over there and down here, involve folds that just quite notably involve many loops. Just also anecdotally, if you randomly sample structures in here, it turns out many of these structures are signaling proteins, which by definition needs to change its conformation for it to function. And in a similar way, the enzyme structures here also possess similar characteristics where many of them are naturally regulated by allostery and they're inherently flexible. Yes.
Is there any prokaryotic versus eukaryotic protein biases? Just because a lot of proteins in our CSBPDB are actually eukaryotic proteins, but expressed in prokaryotic systems.
Yeah. No, we haven't looked at that, but that would be really interesting to see. Yes.
I mean, that, how about the bias protein in the space? Like we probably have a more hydrophilic proteins than rather than hydrophobic, right?
Mhm.
With a difficult to assess. So does it account in this?
It's a little hard to say because all of these representations are backbone only representations. So you strip away all the sidechain information, and this is only looking at topology on the local and global scale of just the backbone architectures. So we don't know really the amino acid identities here at all. Yes.
Are in fusion, but like if there are, or if you try this, like if you train on a subset of the data set, does it have a different bias than what we observe in this main art fusion?
Yeah. That's one of the main things that we wanted to try but didn't. There's many versions like the motif scaffolding version and the secondary structure fold condition version. I think for conditional models like those, it could be you could probably avoid this sampling bias entirely just by steering it towards certain regions. But yeah, unfortunately, we didn't try that for this study. Well, what we do know is that at least for Propardelli, which is the model we've worked much more closely with, there are these kind of behaviors where depending on the parameters, so collapse on some modes of the distribution is actually more likely than not. And so I don't know if anyone has tried Propardelli here, but the original version of Propardelli would only generate tin barrels and nothing else. So now that issue has been fixed, but it was quite a difficult issue to address. Yes.
This protein alkaline and AlphaFold are both all backbone only items.
So yeah, so I guess what I mean by backbone only is that these embeddings that I'm using here are the backbone only representations of structures. And the projections that you see only contain information on the backbone coordinate level. But then when you do self-consistency with AlphaFold, it is not backbone only, but the metrics are assessed on backbone only. So you would only look at like backbone RMSD instead of sidechain RMSD because you don't have the initial sidechains to start with. So just with other representations too, I only showed ESM3 in the previous slide. But you can also look at the embedding spaces of protein MPNN. Here is just the first layer. So right at the first layer was just its immediate 48 nearest neighbors, and a global representation, which is the domain segmenter. And you can notice in essentially all three of these embeddings, we see this dramatic bias in the sample distribution and the ground truth. Again, very interesting that these are also sorted by helicity within the first two principal components. So how do we quantify this bias? So we take inspiration from the image diffusion community, and we essentially take their FID metric, which is the Fréchet inception distance, and also inspiration from the EvoDiff paper, where they define the similarity in the protein language model embedding space. But in here, we're using the embeddings from all of these different structure representations. So the FPD, or we call it the Fréchet Protein Distance, is defined really the same way. So if your mean of the data distribution and your sample distribution match, then it's 0. And the covariances, if they match more closely, then this is a lower number. And the numbers are higher if they do not match very closely. So there's a very fundamental assumption underlying this metric is that both your data and your sample distribution can be explained as multivariate Gaussians, which is definitely not the case. So despite that very serious limitation, it's still a relatively useful metric to use for model development. And you can see that it tracks with intuition. So undersampling and this bias sampling, you have an FPD of 122. This less bias sampling from Properdelli has a FPD of 5. Okay, so we're going to just walk through a lot of figures, so I apologize for that. But essentially, you can just look at the clouds and see if there's more overlap or less overlap. So the color scheme is a little different. The darker dots is the reference distribution, and the lighter dots are your samples. So, but what's noticeable, so each of the columns, so this is chroma, Gini2, RF diffusion, and Properdelli. The multiflow results will be left till the end. And what we're doing here is that we're systematically sweeping over different sampling temperatures. So there's a knob in all of these models that can tune the sample distribution. The top row is your default, which is in all of these cases the lowest sampling temperature for all of these. So it just happens that for chroma, the inverse temperature 10 is their default. And for Properdelli, the default is 1.2. Then as you go down the rows, you increase the sampling temperature. Where you get to the bottom row, you have the highest sampling temperature. And so intuitively, it actually checks out and makes very reasonable sense is that you have highest temperatures sort of matching that distribution more for Properdelli and for Gini and for chroma. But what's quite interesting is that for RF diffusion, that's not the case. As you increase the noise scale, it turns out that these samples, if you look through the rasterized plots in the scan QR code, what you'll see and find is that some of these samples will have still idealized local secondary structures, but they're they're unpacked. Poor packing, despite good local secondary structures. And that could explain this drift in the sample distribution away from the ground truth. Whereas all these other models we've considered, increased temperature, better coverage. So the FPD that's annotated in each of these, it's a little hard to see, but there will be a separate plot later on showing you the trends. it's stratified by FPD-D and FPD-ND. And this is an important distinction to make because one reasonable question is that, yes, you're improving coverage throughout this averaged space of representations, but what if you're just making garbage structures? And designability, so we're not arguing against replacing designability entirely. It's still a very useful metric to guide the design and synthesis of candidate sequences. So we still want to make sure that our samples are kind of designable, whereas we don't want to just say, okay, better coverage equals better model. What's reassuring is that for some of these models, as coverage improves, that FPD-D, that designable subset, the coverage of that also improves. But that trend is not the same for all of these models. So for example, for Propardelli, the FPD-D goes from 51 down to 36, down to 21. But what you'll also notice is that the ND, which is the non-designable samples, that FPD is much lower. So it seems like to just get better coverage, almost you just have to get away with, you just have to accept that a lot of your samples will be undesignable. And that goes hand in hand with that metric we've quoted earlier, which is that statistic we quoted earlier, which was even the native sequences, 40 to 60% are non-designable. So there's still some subspaces of protein structures that perhaps are not passing through the designability filter because of limitations in the sequence design step and the structure prediction steps, or the global alignments that's being imposed. So very quickly, these other embeddings, we kind of see similar trends where you see biases across most of these models as well. And one important thing to know is that the FPD scales are embedding dependent. So these embeddings are with the domain segmenter, and you'll notice that these FPD values are much lower. With the ESM3, they were in the hundreds, and here is in the single digits. But what's really interesting about this protein domain segmenter embedding is that good coverage under this representation space implies that your model can cover all functional cath domains. So cath is this manually curated domain data set where it decomposes large protein structure data sets into these functional building blocks. So good coverage under this embedding space can mean that your generative model can sample broadly and across all functional building blocks of proteins. And the biases for protein MPNN are quite stark, but you'll notice again the scales are much different. Yeah, just some more plots, and this is sort of narrowed down to just looking at Propardelli in the bottom row, where we see that. So the public model was that one I previously mentioned with that collapse on tin barrels, and you'll see that as you increase the sampling temperature from 1.2 to 1 to 0.8. And note that the step scale is actually inversely related to the temperature. So because this is based on this the EDM scheme introduced by Keras, the higher the step scale means that you trust the scores that's learned by the model more. And so that corresponds to a lower sampling temperature. And just as a point of reference, the Boltz model, which is also based on the EDM framework, they use a step scale of, I think, around 1.6. So it's very low temperature sampling for this. Yes.
Like these embeddings all trained on the same data set as our fusion, or like what data sets are we embedding?
Yeah, all of these embedding models are trained on slightly different data sets. Yeah, not the exact same data set. And that's just one unfortunate reality you have to accept, that there's no way to rigorously do that unless you retrain these yourself. Okay. So for protein MPNN embeddings, again, you see the biases, layer 2. So I didn't show layer 2 before. But again, with layer 2, there's similar biases, and also with layer 3. Okay, so now we've seen that sample distributions tend to be biased, but what if the ground truth itself is biased? And that is a common argument that Kevin Yang makes in his papers, whereas because most proteins can't actually be crystallized. So the PDB is actually itself a biased distribution. Um, so the PDB can be thought of as the observable protein structure universe. And it's self-biased because de novo design fundamentally argues that there is a much larger space of realizable protein structures that can be viable with design sequences that's beyond the currently observed set. And that's indeed what you see empirically with the generalization you see from RF diffusion. But again, there are additional biases coming from the inherent limitations of these experimental techniques that determine structure, and also just of subjective researcher interest on determining the structures and spending time and effort on some proteins, but not others. So we have this kind of trade-off, and I think it's worth discussing that perhaps, we don't need to cover all of ground truth, and that's the avenue that some models do strive for, like RF diffusion, where you can think of RF diffusion as a highly regularized model where you have these highly idealized substructures with very few loops. And effectively, this is removing structural vestiges of evolution, where there are these sequence and structural liabilities that hamper expression and yield that is really the pain of crystallographers that are no longer the case when you idealize by sampling from this temperature-adjusted, very peaked distribution. Or you can do more interpolation with more loops. So this is an avenue that other models might strive for, is you want to cover everything that's observed in the functional protein universe. And so you don't want to just discard a whole region where you have a lot of enrichment in loops. So I briefly talked about structural quality control, and there's other thresholds. So, resolution, are free, and no chain breaks. In fact, these three alone remove quite a lot from the ground truth data set. So the total size of cath is only about 20,000. So these steps, these are independent. So these are not like just removing 4,000, because then if you add these together, you don't have a data set anymore. So these are just independently looking at, okay, what fraction less than 3, what fraction less than 2.5. And after removing these, you're still left with kind of a sizable data set, but it does remove quite a bit. So the question of what if your ground truth is biased? Well, let's just change the ground truth to another distribution. So we did cath, which was the first ground truth we've tested, and we switched it up. We switched cath to the PDB. So this is a cluster version of the PDB, and these are not split by domains, unlike cath. And again, with a different ground truth, we observe similar trends in the FPD. So this is just to confirm that the trends we observed with that cath as the ground truth is real. Yes. Yeah.
What is the R-free metric?
Oh, yeah. So this is it's called the free R factor, and when you do fitting of your atomic coordinates, when you get X-ray crystallography data sets, there is essentially you're doing like a data fitting kind of procedure. And before this R-free metric was introduced in the 90s, what people do is they try to fit atoms to explain electron density on the whole data set. But by doing that, you could artificially improve your fit, but essentially overfit to your crystallographic data. And so R-free is essentially the correlation or how good your fit is of the atomic model to the crystal data on a holdout set of your structure factors or your diffraction patterns in crystallography.
The structural vestiges, like is it just a matter of the fraction?
Yeah, I think there might be some very thoughtful review papers. Like really, this is an opinion from me. It's really so, I guess my opinion on these structural vestiges are these effectively, you start off with some ancestral sequence, and those are kind of carried forward as you make mutations and may be detrimental to stability. And evolution doesn't explicitly tune for thermal stability or yield in E. coli. So these are these so-called structural vestiges as any of these factors that hamper expression and yield. Yes.
I think you said it, but did you say it on the like 20,000 different? Okay.
Yeah, in the cath data set, yes. Yeah, or different domains. Yeah, so this is just with the PDB as the reference, and you see these similar biases. So some of these are actually quite dramatic. You do see non-overlap in most of these models. Okay, I think we're just a few more slides. The coverage analysis is great, but we still kind of want to ground ourselves in designability, because that's still the de facto metric by which experimentalists test and filter computational designs. So while it is true that increasing sampling temperature gives you better coverage, which agrees with intuition, you also see this trend of increasing sampling temperature means worse designability. And this trend actually is consistent across all models for which we sweep over the temperatures. There's a caveat about Chroma because they have their own sequence design model, which is not ProteinMPNN. And it's been shown also by Sergey in his tweets that if you use Chroma design, it's much better on Chroma backbones. And ProteinMPN does not really like Chroma backbones. So these values could actually be much higher if we switched the sequence design model. So here's just a summary of all of those scatter plots I showed before. These are FPD metrics calculated with ESM3 embeddings using designable samples only, so just the designable subset. And in terms of ranking, so again, you see that higher sampling temperatures with these diamond entries here generally have lower designability across all of these models. And the coverage of native structures in the native cath distribution, there's a ranking of Properdelli has the most faithful coverage of cath, followed by Genie2, and then Chroma, and then RF diffusion, at least under the ESM3 embedding scheme. But what's interesting also, I've kind of alluded to this before, is as you increase sampling temperature, that does not always mean better coverage. So it does imply better coverage, this downward left slope for Properdelli. But actually for, in this case, Chroma and RF diffusion, higher temperatures actually meant worse coverage. Okay, so this is kind of an impossible figure to read, but I'll zoom into some of these details. Essentially, what I'm looking at here is, this is sort of just like a side analysis, but there's one interesting nugget in here that I think would be interesting to comment on. Here is the fraction of your samples that's designable as a function of what cutoff you use for a self-consistent RMSD or a self-consistent TM score. So literature always says 2 angstroms for MSD, or maybe 0.6 or 0.7 or 0.8 for TM. So it really varies. But what you find is that I also stratified by if you only use the self-consistent metrics, or if you also add on a PLDDT metric as part of the designability criteria. So what you see for multiflow is that that gap between adding a PLDDT criterion is quite small. So just by adding a confidence filter, you don't actually drop the fraction by much. But that gap is massive for Properdelli. So it suggests that a lot of the samples coming out of Properdelli are low confidence, but it actually has the right RMSD metrics. So that's just one comment, and this could potentially suggest some biases in the PLDT metric and whether or not to use that as part of evaluation. Because we also do know also anecdotally that fewer secondary structures imply lower PLDT and inverse. So higher secondary structures, in particular alpha helices, imply higher PLDTs. Okay, and then finally, these are fold-seek tokens. And we observe biases across fold-seek tokens as well. So fold-seek is this discrete representation of protein structure. You have this discretized alphabet of 20, and we're essentially counting the frequencies of the 20 alphabets within each length range bin. So these are these unigram frequencies, and we're computing the KL divergence between the frequencies of your sample distribution and the frequencies of your native distribution. So we see indeed that actually there still is quite a bit of bias, but the trend is the same, right? As you increase sampling temperature from the dark purple down to here, we see lower KL divergence. And so this checks out with these discrete embeddings as well. But stratifying it by length, you see also different behaviors from all of these models. Whereas for RF diffusion, these peptides are primarily all alpha helices. It's very rare to actually ever get any beta hairpin peptides of less than 50 amino acids. And that explains this really sharp peak up here. But in general, you do see the same trends with continuous embeddings here. And finally, for completeness, we have multi-flow, where you also indeed see that de novo streak, as you saw with RF diffusion and this bias sampling. So what is interesting about multi-flow is that they train their model on an protein MPNN redesigned version of the PDB. So they basically train on designable samples only. And by doing that, you're shifting that model distribution at the training step already. So you're already modeling this bias distribution. So it's a kind of unsurprising that you see this massive distributional shift across all of these embeddings for multi-flow. And for interpretability, the secondary structure distributions also have similar trends. But again, it differs by model. So for example, Properdelli, as you increase the sampling temperature, you get a better match of the native secondary structure distributions. But that is not always the case for these other models. So, with Gini 2, as you increase the scale, the gamma parameter here, you get less of a shift towards the beta sheets. You can also, this is going back to one of the earlier questions about functional coverage and whether or not there's biases and what kind of functional domains are being covered. So this, I guess, so how would you read this plot is looking at these two down here, where you have distribution over all of the domain classifications, architecture classifications in Cath. And you'll notice that there are some missing bars in here, and also some missing bars in here. So the missing bars in the sample distribution, in particular, let's say Gini 2 at the default noise scale, indicate that out of 22,000 samples, it is not generating the domain that actually was present in Cath. So instead of looking at it from an embedding perspective, this is predicting what architecture each of these sample structures have, and essentially recording their frequencies and seeing what kind of architectures are completely being missed out and oversampled, vice versa. Okay, so final slide, and then we can maybe open for questions. This is the last main figure in the paper, where sort of the hypothesis is that if Properdelli actually covers the distribution very faithfully, we should be able to find these native structural elements in Properdelli more often than the other models. And these structural elements are taken from terms, which are these tertiary structural motifs from the Gregorian lab. And these exist in the PDB. So these are all metal binding sites. And we just took these functional sites as this test to see if they occur. So going from left to right, we have several bars. So the leftmost bar in dark purple is how many times do each of these substructures occur in cath. And that gives you a reference of kind of saying how many times do you expect these motifs to occur in the ground truth distribution. Then going from left to right, we have chroma occupying the next 3 bars, and then Gini 2, the next 3 bars, multiflow for 1 bar, and then RF diffusion. And then the final from the right, the 3 bars from the right are Properdeli. So for some of these motifs, actually many of the models do not recapitulate the natural frequency. And it's actually quite surprising for Properdeli because Properdeli was trained on cath. So perhaps you could expect it to match the cath frequency, but some caveats here is that this is unconditional sampling, so we're not doing motif scaffolding in any of this. So sampling might be limited because we're only drawing 22,000 samples. But what you'll notice is that for some of these motifs or some of these terms, where most models struggle to cover, you do see non-zero counts from the library of scaffolds or unconditionally generated by Properdeli. So you see non-zero bars down here in the right 3 bars, and essentially for most of these as well. Okay, and maybe this one more slide is that there's some very minor details on the PDB. I don't know if anyone has tried training like a generative model with relative positional encodings on the residue index provided by the authors, because there's wild inconsistencies in the residue index provided by the authors. And so I'll just really quickly go over these slides. I think it's really interesting. And I don't think anyone really talks about these things in papers. So basically the problem is a lot of examples in cath have mismatch between the residue index and the actual gap size. So the model will think that there's a gap of 3, but or the model in this case will think there's a gap of over 1,000, but in reality, there's only 3 missing residues. And the residue indexing is assigned by the authors through a series of conventions either in their field. It doesn't have to be monotonically increasing even. So it could go backwards, it could be negative, it could skip 1. So there are cases where it just skips residue indices entirely. So one of the tricks is to actually just keep this fundamental property where 2 residues have consecutive indices if and only if they are peptide bonded. And by keeping this property, you actually can train models much more stably. Yes.
Small comment, but this has been fixed in MMC files.
Yes.
So
yes, you're correct.
Deprecated format.
Yes.
Don't use PDB.
Yeah, so that's a good point. If you use BioPython, you load in the residue index. If you load in the label seq ID from the CIF files, you don't encounter any of these nightmarish issues. Yeah, so here's just like some examples of here's a case where the unresolved residue gap is only this much, but in your sequence, you have huge jumps in the residue index. And here's like a nice Protopedia page kind of saying there are cases where there's a jump in the residue index, but your backbone's actually fully connected. And same thing down here. There's a jump, but it's fully connected. And then there are also these really weird edge cases here that's really important to filter out from your data set, especially when you're considering something like training your model on these distributions. Oh yeah, and insertion codes are a nightmare as well. So yeah, finally some discussion points. the non-designable native structures, while that might seem like a flaw of some of these models, perhaps it might be a feature in that the space of protein-MPNN and the space of ESM fold that can pass these designability filters are already highly regularized. And so for the purpose of de novo design, this is a good thing, that maybe you don't want to spend so much resources trying designs in a region that doesn't have as high of a success rate. So even though we tried to correct for missing residues, there's still crystallographic artifacts that might be possible in the ground truth distribution. So potentially one way to remedy this is to standardize the crystallographic refinement procedure through something like PDB redo, or essentially doing a refolding of everything in the PDB. Different combinations in that matrix of what sequence design model to choose. So right now we only really look at protein MPNN and ESM fold, but you can also do the same thing with like chroma design, omega fold. Many of the later models like bolts or and so on, they might lead to drastically different designability conclusions. And similar to the philosophy of like a newer work in this field, which is Proteina, an ideal model might actually just be something that is steerable. So maybe it's fine that unconditional samples don't cover your full distribution broadly, but as long as you're able to steer and guide through reconstruction guidance or other RL-based fine-tuning methods to a certain desirable subset where you can actually get functional designs, that's really all that matters. And again, these are all unconditional samples. So conditional FPD might give drastically different conclusions. Many models are not benchmarked. So Plaid from Peter Abil's lab here and many other models we haven't considered. We only picked some representative models to benchmark. But all of these models, many models could have their own distinct biases and behaviors, and they're all optimized in their slightly on different ways, and they're all trained on their different data sets. So we're hoping that the shapes analysis is not just analysis of just these 5 models, but a kind of a framework to apply to the analysis of any future models that will be coming out in the field. Right, so we only considered backbone-only features, as we mentioned. Now with more all-atom models coming online, these sidechain aware embeddings would become increasingly important. So, for example, ligand MPNN, for example, that is sidechain conditional with their features, or even protein language model embeddings of the design sequences from these inverse folded or sequence design distributions of all of these sample distributions would be interesting. So we only considered backbone structure distributions, but there are other distributions from which perspectives could be really interesting. And through that analysis, this is just like very recent work, is that through that analysis, we were able to tune Proberdelli and find a right setting which has broad coverage and that can be conditioned and steered so that we can do effective motif scaffolding. And so this is a very recent result that we haven't released yet, but on the motif benchmark, which some of you might be aware of, this is like given the motif, generate the scaffold that will hold that motif in place. We are seeing some early results that suggest we do outperform RF diffusion and are able to competitively solve some of its tasks that it can already solve, but most crucially also solve tasks that were not solvable previously. So with that, thanks everyone for your attention, and thanks Melissa. And I've all my Yes.
I was wondering, it seems like a big concern with benchmarking fully like in silico is that even your tests, you're relying on protein MPNN and its biases and AlphaFold and its biases. and I'm not too familiar with the space, are there physical simulations that, so we can exchange computation for lack of bias somehow that you can use to verify these hypotheses?
Yeah. I think right now the consensus is that even though some of the predicted structures might have physical violations, simulations themselves, like molecular dynamics, have their own problems and their own errors. So there are lines of work trying to match the sampled structures from these models closer with simulated structures coming from like a physics-based force field. But I think ultimately, really the test for designability is going to be something that is like a high throughput assay and not anything in silico. Yes.
Do you know how protein MPNN kind of became like the de facto sequence design model in this pipeline?
Yeah. That's a good question. So around the time Protein MPNN was released, there were other neural networks for sequence design that had even higher sequence recovery, right? So there were models like 3D CNNs from Ross Thayer's lab and like the mute compute servers, and also like the previous precursor to Protein MPNN and others. I think it comes down to experimental validation. I'm sure other models have been tried and tested as well. But it's just that the scale of experimental validation for protein MPNA is very convincing in that they were able to show they can rescue designs that failed with Rosetta design quite significantly. So, and also they were able to show, I think probably like figure 2 of like protein MPN's paper, where they show like the single sequence AlphaFold structure prediction to be really accurate when you redesign your sequence with protein MPNN. That might have like spur on these thoughts of, oh, now you can use AlphaFold as like this in silico designability filter in single sequence mode. And protein MPNN can kind of let you jump to those solutions really quickly. Yes.
Fine-tuning approaches are like, I guess in an image context, people use like LoRa's or things like that you can apply to try to bias the distribution in a direction that you're interested in.
Yes. So one of the works that I mentioned in this like big slide here, Protina, this one, they apply LoRa to fine-tune their model. And yeah, they're able to steer it towards. So I think they first train on everything, both undesignable and designable samples, and then they fine tune with LoRa towards the designable subset.
You know, when they fine tune, they can get into the like kind of gap spaces that you were highlighting, or are they just kind of like focusing it within the distribution that already exists?
Yeah, I think, well, I guess to answer that, I would have to benchmark their model under this. I think from their results, it seems like, so from my understanding is that they fine tune on natural or native structures that are designable. So even though while they've trained on everything, ultimately the final model ignores the non-designable subspaces. So it would be this like sliver of all structures that they're focusing in on, which is fine, I guess. Yeah, if that's your goal. Yeah.
I was curious, if you'd ever just thought about sort of specifically benchmarking, like, like very, like, specifically looking at proteins for, I don't know, they're like top, I don't know, like only membrane proteins in certain cap families or such, and just looking at the coverage and your sort of replicate the same analysis on like proteins, so there's like just like but it's certain models together.
Yeah. That would be really interesting. I think like to the extent of that analysis, this is sort of all that we did was that we we looked at the domain architectures of all the PDBs that were classified as enzymes. And I guess yes, you could definitely do the same thing with those that are annotated as having some allosteric mechanism. But yeah, I guess yeah, anecdotally, the region that was not covered up here by the ESM in the ESM embedding space has a lot of structures that are allosterically regulated. So basically this region here, if you just pick a random sample and look up the PDB, it turns out these are mostly signaling proteins. Yeah.
I have a question about this kind of like a discussion kind of question. So they released a new like steering mechanism for balls that uses like test time inference for the steering. And they are using some kind of constraints to, let's say, prevent some clashes after they like predict the structure. So do we think in the protein design domain, we know enough constraints that we can use test time inference and stability in order to get rid of the bias from these models? What do you think about this?
Yeah, that's a really interesting question. I guess it's not directly so in their case, their clashes are all of these physical priors are very well established. Yeah. So here we don't really know what are the designable spaces of architectures.
But
Um,
if you like, if you want to design or like introduce a constraint, let's say, what would you choose? Like,
Yeah.
you have
So I think in terms of like the Rosetta energy function, that's still quite, even though like using it for filtering is not has a low success rate, there are components in that energy function out of those 19 different terms. There are useful pieces of that where you can look at a structure and make a hypothesis in saying here is a region that you don't see in the native distribution. And because a lot of the terms in that Rosetta energy function is fitted on statistical frequencies in the PDB. So yeah, I could imagine having a kind of tuning setup where you use those terms to guide. Yes.
Question going back to the temperature parameter.
Yeah.
That are you talking about on average it decreases designability because the point is that it adds diversity, right? So you might end up with one sequence, for example, that is actually a better, more stable sequence and fits your backbone better, even if you have that higher temperature. It's just that you have to do it over and over and over again to find that one.
Yes. So in practice, like if you want to sample from a region that if at a lower temperature you never reach, then increasing the temperature while you can accommodate that low success rate in silico, but you're able to spread out your distribution more. I think that's a valid trade-off to use. And that's in practice, that's some of the designs I've synthesized before are from that strategy where that hit rate is very low, but because some of these models you can sample pretty quickly, you can generate a few 100,000 samples pretty fast. So you're still able to get those designable samples. Yes.
So, that's a good question. So, increasing the designability of structure space is not necessarily gives it this to go better model because you said it's increasing the garbage structure. So, what can be kind of examples that be garbage?
Yeah.
Is that the structure I haven't seen before it is not in because it's not in cat, or because we probably removed the ones that are physically not impossible, right? But if we leave those down, what are the left ones that are can be seen as garbage?
So I think, that goes back to the idea of like what counts as a success, or what counts as designable. I think in the end, as long as your protein is able to be expressed, and
Wow.
or even if the structure hasn't been found and could never be found, even if it's like a disordered protein, as long as it can be exists in some buffer condition, and that is not just aggregating or cleaved by proteases, that's a valid protein. A lot of these designs might not be, and I don't want to be picky on things, but like for example, this region down here, I guess by eye you could say that these structures are not designable and probably won't exist in solution. Because after you map these structures back to the sequence space, most of these are going to end up like polyalanine or polyglycine designs. And so those would be the designs that you would want to avoid. But you can see that like this region covers its own space in this embedding space. So it could actually boost the coverage metric. So that's the part where I wanted to avoid, where like you want to make sure that you have both good coverage and these structures are still good. And so yeah. Yes.
Is protein MPNN like it can be conditioned on conditions like pH or temperature, other things like that? Like how, I guess, flexible is designability to like you want to design a protein for a specific like solution or specific condition?
Yeah, that's a great question. I think out of the box, you cannot condition it on pH, but there are attempts and works to adapt protein-MPN to other data sets. Like I think the data set from Gabriel Rocklin, where you have a lot of thermostabilities, that can be something that you could fine-tune protein-MPN on. But I think that's still a really tricky question, or like, and also very valuable to have, is sometimes you want your proteins to function at very low pH levels, and that's actually the project I'm working on right now, is how do you design sequences that survive these very low pHs. It's not entirely, you basically would need to curate a data set of experimental condition to the sequence pairs.
Is there enough data out there on like a lot of these conditions to actually like train a model robustly?
I don't know. Yeah. If anyone knows about that, please let me know as well. Yes.
Do you think Protina as like classifier-free guidance can kind of solve this problem by filling in the gaps, or do you think there's more to those like embedding differences than just like those classifications present in cat?
Yeah, definitely. I think so I guess some of these results down here for Bordelli. So these results for this model use classifier free guidance as well. And so it is definitely a very powerful mechanism to guide generation. And so here like CFG1 means classifier free guidance scale of 1. So that I think that's why that's like one of the main limitations here of this analysis, is that we never really looked into like conditional FPD. So I'm fairly confident that like a lot of these models that can respect conditioning information, you can just navigate into these sub regions of the space that weren't sampled. Yeah.
How
Yeah.
computationally expensive is it to incorporate embeddings for the whole PDB?
Yeah. Oh, yeah. Very good question. So depending on the embedding, so probably like half an hour for ProteinQure, and ESM3 slightly longer than that. And this one is super fast. It's like really lightweight CNN, so less than half an hour, I think.
Think about the SM3. Like,
Mhm.
I remember you said you were taking this embedding directly from the variation autoencoder, right?
Yes.
Did you try to take directly from the model? Because they have different tracks in
Yeah.
the SM3, so see if it's making any difference.
So as I was preparing this presentation, I thought, why didn't I try it from the model?
Because it has, like, only information
Right.
embedded.
Right.
It can at least steer the bias away from the from the VC from variation autoencoder.
Yeah, that could be true. I think the reason why I did that because it's just I already had one representation that was global, and I wanted like another local version. So
Maybe it might not be fair to other models.
yeah.
I think it's 8:30 now, so we'll probably wrap it up, but do you mind maybe sticking around for a
Oh,
few
yeah, for
minutes?
sure.
People have individual questions, but I think we'll call it there. Let's give another round of applause. Thank you all for coming.
Thank you.